It's an absolute privilege today that we have Dr. Stefan Jakobs with us. And I think most of us know he's very much at the forefront of super resolution microscopy. A little bit about Stefan. He studied biology in Germany and the UK and obtained his PhD in 1999 in Cologne. He recorded his first super resolution image using STED already in the year 2000, and at the time he was working as a postdoc in the lab of Stefan Hell, who of course won the joint Nobel Prize in chemistry for his super resolution microscopy. Stefan now heads a research group at MPI for biophysical chemistry in Göttingen, and he is professor of high resolution microscopy at the University Medical Center. Stefan's research focuses on the investigation of mechanisms that determine mitochondrial inner architecture in health and disease. And his lab uses a wide variety of microscopy techniques, including STED, resolved, and brand new MinFlux nanoscopy. So I'm sure Stefan is going to absolutely amaze us today. So I'll pass over to him. Just before I do, I will say, if you have questions during the talk, maybe put your question or even just your name in the chat function and we'll come to them at the end. But Stefan, thank you so much, and it's over to you.
Hi, Kate. Hi, everybody. Thank you very much for this kind introduction, and thank you for having me in this series. I try to share my screen. Hopefully that works. So you should now see my screen. Otherwise, please shout. I'm going to allude a bit on our work on using microscopy, electron microscopy, and super resolution microscopy to study architecture of mitochondria. By the way, here on this collage, you can see on the right-hand side and in the middle reconstructions of FIBSEM data and electron tomography data. And on the left-hand side here, that's a 3D STED image. I'll focus mainly today about our work using super resolution microscopy and will only briefly touch upon the use of electron microscopy. Well, we're interested in mitochondria. Well, why mitochondria? Mitochondria are multifaceted organelles. They obviously have a number of biochemical functions, most notably ATP production. The mitochondria in our body, they produce around the body weight every day in ATP, which is a massive metabolic load. But in addition, there are many other essential processes, including iron-sulfur cluster biogenesis, and of course things like apoptosis and calcium homeostasis, and many other things. Mitochondria can perform all these tasks because they are highly compartmentalized. They do have a rather smooth outer membrane, and then they have this highly folded inner membrane, which forms the cristae, these invaginations here. These cristae, they contain the respiratory chain complexes, whereas the outer membrane mainly contains transporters as the inner boundary membrane. This is this part of the inner membrane which is paralyzed in parallel to the outer membrane here. Where the cristae start to unfold, these sites here, they are called cristae junction. Well, although this textbook image is of course correct with the outer and the highly folded inner membrane, the details can be very, very different. Mitochondria are certainly not always bean-like in a cell. They very often form extended mitochondrial networks of tubules. And when we look to the interior, to the cristae architecture, we can also see a wide variety of different shapes, ranging from, for example, this very densely packed lamellar cristae we see here in this secreting cells to triangle-like cristae and everything in between. Even if you look to more, say, common mitochondria from cultured cells, like here from human cells, a HeLa and a kidney cell, or a cell from a monkey cell line, we still do see difference in the cristae architecture. The density of the cristae might differ. We have lamellar cristae, we have tubular cristae. And we can note that when we grow the cells differently, also the architecture of the cristae adapts. So there is certainly a correlation between form and function. Also, we do not really know how strict this correlation is. And certainly the players that are involved in changing these architecture might be known, that it's the ATPase, that is OPA1, that is the Mycos Complex, that are lipids. But how the interplay of these players is and how they are regulated to changes in the architecture of mitochondria is still largely unknown. And one reason for that might arguably the fact that we do need microscopy to study on the single molecule level the architecture of the mitochondria. We need to study where are the proteins and how do they interact on a molecular scale. Well, for that we do need microscopy. Mitochondria are small, and for that reason I would argue that we do need quite a lot of different technologies to study those. Network morphology obviously can be done with diffraction limited microscopy, confocal microscopy, for example. Whereas if you want to look into the mitochondria, if you do want to see individual cristae, we do need higher resolution, we do need super resolution microscopy. For some mitochondria, we indeed need subdiffraction resolution microscopy or nanoscopy, as I prefer to call it, such as STED microscopy. For others, when the mitochondria are a bit larger, then things like ZIM, structural elimination microscopy, is certainly sufficient. So in that sense, this cartoon here is a bit misleading, because one really needs to look what mitochondria we're looking at. And for the highest resolution, of course, electron microscopy is still the gold standard, although with the newest approaches, such as min-flux nanoscopy, we are now in the single-digit regime for localization precision, and of course can also localize here with very high precision molecules to structures within the mitochondria. So what can we really see here? Well, just as an example, that's a human cell line labeled for 4 dyes. We have the nucleus, we have the actin and the tubular cytoskeleton in gray and green, and the mitochondria in fire color. So that is this extended network which goes through the entire cell. If you zoom in into one of these individual tubules here with the confocal microscope, then we are not able to see any substructures. And that is just, of course, due to diffraction. These mitochondria have a diameter of around 300, maybe 400 nanometers. And confocal is just not sufficient to visualize structures within these organelles. However, if you use STED nanoscopy, then we see already here in this representation that there is a substructure, and if you zoom in a bit, then we start to see, and here's another zoom, we start to see that indeed we can see different protein distributions. So what we've been labeling here is with red the ATP synthase, and this is enriched at the cristae membrane. So we do see in this chemically fixed cell here indeed individual cristae, and in green is a protein called MYC60. This protein is part of the Mycos Complex, which is enriched at the cristae junctions. So what we are seeing here is a lamellar cristae, presumably a lamellar cristae, and 2 cristae junctions marked by the Mycos Complex. I should say that recording cristae in fixed cells is a challenge, and it's a challenge because fixation is certainly not very easy to maintain the structure, and also the protein density in these structures is not very high, so it's not easy to generate this kind of images in fixed cells. It's much simpler, apparently, to do so in living cells. So we are looking here to a transgenic cell line we generated a couple of years ago, which overexpresses COX-8A, tagged to a halo tag, and then we labeled in a living cell these cells. Here on the left-hand corner, you can see the confocal image. Here on the right-hand side, we do see the mitochondria, and you can see, hopefully you can see it here, the individual stripes which represent the cristae. To show that it's really cristae, we can record large images. We can zoom just to individual parts and make intensity histograms across these stripes. And what you see is that the distance, peak-to-peak distance between these individual stripes is around or less than 90 to 100 nanometers or so. And if we record an electron microscopic image of the very same cell, then we are in exactly this size regime. So what we are seeing here are indeed individual cristae. And it's live cell, so while we can see the mitochondria moving. So here we have been labeling individual cristae, and in red that are the nucleoids. So in this cell type here, it looks like as if there are groups of cristae which are spaced with nucleoids, and I will come to these groups of cristae later in my talk again. When looking to these images, one can have, of course, several questions, and my lab has two major questions, I guess. One is, how are these individual cristae formed? How do they develop? And then, of course, how are they degraded and replaced? And the other question is, why are the mitochondria tubules? The latter is not a so really simple question, because if we modify a number of, or if we delete quite a few proteins in say, for example, yeast, then the mitochondrial skeleton, or the mitochondrial network, which you see here in a typical budding yeast cell, which is again a very well elongated and extended, collapses, so that the mitochondria do form individual large spheres. And the same happens if you purify mitochondria, then the mitochondria collapse into spheres. So there must be something which keeps them in shape. And the question is, what keeps the mitochondria in shape? This is unknown. People have been speculating about something like a mitochondrial skeleton. Well, this is certainly not a microtubule skeleton, and certainly not an actin cytoskeleton within the mitochondria. We know that is not existing. But what is existing in bacteria is bacterial cytoskeletons. They are quite diverse, can be made out of different proteins, and of course mitochondria are derived from alpha proteobacteria. So one may ask the question, is some related structure already existing in mitochondria? And if yes, what could that be? And I would like to touch a bit on this question for the next couple of minutes, although we have maybe not a definite answer to this yet. A very interesting protein complex, which is very much involved in the maintenance and the generation of the structure of mitochondria, is the Mycos complex. The Mycos complex is a protein complex which consists in mammalian cells out of about 7 proteins. It has been found already 10 years ago independently by 3 groups, by the groups of Neupert, Nonari, and van der Laan. And it has been found by different means, and the group showed that this protein this protein complex is required for the overall structure of the mitochondria, but it's also required for the maintenance or the generation of the cristae architecture. In one of these three papers, Julian and Mary made a very interesting proposal, and that was that the complex might be required, and she already showed data using fluorescence microscopy. In that work, they showed that the Mycos complex might form something like a spiral-like structure in the mitochondria, which might be required for the architecture of the mitochondria. So we thought that is something we should look into it and use super resolution microscopy to look at the distribution of MIC60 in mitochondria, and we started with yeast mitochondria. So here is a Saccharomyces cerevisiae yeast cells expressing MIC60 GFP. In the yeast, the fusion protein is functional. And this is a yeast cell grown on glucose, so it has this ring-like structure here. With confocal, basically nothing, no substructure is visible. However, with STED microscopy, then once in a sudden, one can often see structures like this, where we see individual clusters that are MIC60 clusters. But I hope you're with me and agree that in this very case here, these clusters seem to form what we believe or what looks like, a helical structure, something like a helix or so. So there seems to be an underlying structure in this yeast cell. I should say we see it often, but we don't see it always, and it's not always that beautiful, which might indicate that if this is a structure, it's highly dynamic and it might have very, very different flavors. So we also looked in other cells, in human cells, whether we can find such a similar structure. Here we are looking to a human cell line, a HDFA cell line. With a STED microscope, I zoom in into these two parts here. And if I do so, again, we do see clusters of Mycos, of MIC60, sorry. And again, I would argue that we do have here two bands which seem to be twisted in this mitochondrium. So this here is a mitochondrium, and in the red we do see here these dots which seem to form a structure. So the interpretation of this data here would be that in this very case, we do have mitochondria where on two sides clusters are, Mycos it's in a boundary membrane, which are opposite of each other, and sometimes that we then have a twist. We also looked in other cell lines, whether we can find it. This, by the way, here is 2D STED microscopy, so we also looked at 3D. And if I switch on the movie, I hope, although this is Zoom and the data is of course compressed, you are able to see that also in this cell line, there seems to be such an arrangement that there is a twisting. We looked a bit more careful into that and did quite a bit of analysis to have an unbiased view. So the idea, of course, is if it is right that the MIC60 is at two sides of the mitochondrial tubules, like it's shown here in this model, then we should, like in this model, then we should get an image like this, whereas in case of the, for example, an outer membrane protein like TOM20, which should be randomly, or is expected to be randomly distributed on the mitochondria, we should get an image like a ring, a full ring-like structure. So we looked into that. Here's one example that is measured data. So we averaged here, accumulated data for, I think, a micrometer along the axis of the mitochondria, and for the TOM20, we indeed find these ring-like structures, where for the MIC60, we indeed find examples where they are opposing. To have a more comprehensive view, we did that for very large data stacks. I don't want to go into the detail here. But what we found is that it was an unbiased computer-based analysis that indeed we find it more often, or very, very often, that the TOM20 is homogeneous distributed in a ring, whereas the MIC60 is not. And on the other hand, we often see that the MIC60 is distributed on two bands, and the TOM20 almost never. So that is quite solid support that we do have indeed this situation here, that the TOM20 is homogeneously distributed, but MIC60 forms these two bands, opposite bands, sometimes twisted. Well, we don't see that everywhere and in every cell type, and I will come later in my talk also to some mutants which can generate these tubulars. But we can make a number of predictions on if MIC60 would be a part of a skeletal structure. We would expect that these individual clusters here are indeed connected. We never see a large filamentous structures of MIC60. So if it is a larger structure, it should be connected. Now, the MIC60 is a promiscuous protein from biochemical data, so we know that it's interacting with very, very many different proteins. So it might well be a platform for other proteins that are then recruited to the MIC60, and that together they might form such a structure. So the prediction would be, if we look to other proteins, they also should form such a pattern structure. And indeed, for some we do that. I just want to give you one example. Here's again the MIC60 structure. If you look to the very same cell, in this case to TOM, so it's to porin, an outer membrane protein, we often also find a pattern structure. It's not the same, but it's very intriguing that we do see for many proteins, porin is just an example, such a pattern. So the idea that we are proposing is that MIC60 together with other proteins might form such a long multi-protein structure in the mitochondria, which is required for having tubules. Well, there is one prediction one can make, and that is if the MIC60 is indeed an helical structure, then we should see a spiral-like arrangement of the cristae. And indeed, that is something what we very often see. It has also been reported for other mitochondria, for example, in flies. I'd like to just focus here on our data on the yeast. So that is a collaboration with Saskia Lippens in Ghent in Belgium. They recorded 15 data of yeast mitochondria. And if I start the movie, and again, I hope Zoom allows to appreciate the data appropriately, then I hope that you are able to see that also in these cristae here, there seems to be a spiral-like arrangement, very much indicating that this is due to the spiral-like arrangement of the cristae planktons. And well, one can quantify that, of course. Okay, that's the rest of the movie. One can quantify that, and if you do that, we find that not in all, but in the majority of our data sets, we do see this propeller-like crystal rotation. So we would argue that MIC60 is not, and this is very solid data, that MIC60 is at the crystal junctions, that along a mitochondrial tubules, it's distributed often in these opposite bands, interacts with many other proteins, and together they potentially might form a structure which is required for maintaining the structure. Well, to look into that in more detail, we presumably need to understand in more detail the detailed orientation or the detailed distributions of subunits of the Mycos complex. So we decided to make use of just recently developed techniques, such as min-flux nanoscopy, which has here been developed at the very place where I am now, and Stefan Health Department in Göttingen, and my lab also has a min-flux microscope by now, and try to investigate sub-mitochondrial localizations of these proteins. We are very at the beginning, so we are really just now exploring the possibilities of this technology, which provides a single-digit localization precision in 3D. So that's really the first beginning of what I can show here, and it's rather still preliminary, but I would like to show you some data. So we labeled cells, in this case here, HeLa cells for 2 different or 3 different proteins of the Mycos Complex. We labeled the MIC60 and the MIC19, and at the same time MIC60 and MIC10. And what we find, if we look to these data sets that are typical min-flux data sets, it's of course a single localization microscopy data, so an individual points here, and it's very difficult by eye to come to any conclusions, but we can analyze the protein, or the distance, the closest distance from the individual localizations, and if you do that, then we find that for the MIC19, MIC60 pairs, we find on average a very relatively narrow a distance of around 20 nanometers, and for the MIC10 to MIC60 pairs, it's around 50 nanometers. I should say here that these are fixed cells, and we have been labeling them with primarily labeled antibodies. A bit more maybe interesting is if we look to the variation of the distances, and there we find that the MIC90 to MIC60 variation is pretty low, whereas we do see a lot of variations in the distance between MIC10 and MIC60, and that kind of fits very well to more recent biochemical data that shows that we do have a MIC60 and a MIC10 subcomplex within the Mycos supercomplex. So MIC60 and MIC19 form a subcomplex, fitting very well to the closer distance here, and the relatively rigid variability, whereas the MIC10 and the MIC60 subcomplex may able to dissociate, which reflects the longer distance and also this variation here. So we are just beginning here to study this data, but I would believe that there's a tremendous potential in exploring this kind of resolution for mitochondria. What you may have also seen here is that in this cell line, in this very HeLa cell line here, there are no MIC60 distribution bands. At least they are not visible as such. So we decided to look with a different technique also to this. And here, again, sorry, here's, I wanted to show that here is a 3D data set of the min-flux data I just showed you. That's the MIC10 and MIC60, I suppose. And there are just no bands visible. So we looked at that with a different technique. And the other technique is 4-pi storm. And here all the credit goes to Mark Bates, because he built a microscope and he recorded also the images. And Mark started to look to cell lines labeled for MIC60, and he looked to a U2S cell line first. That is here on the right hand side. That is a cell line where we also didn't see with the min-flux really structures. Sometimes we had the impression, but not really. And then he also looked to a COS7. This is a monkey cell line. And here in these projections of the 4-pi storm data, it looks also here that, like in the min-flux, we don't see any structures, whereas in the COS7 data, you might see in these projections here some rim-like arrangements of the MIC60 data. So let's look to the 3D data sets, animated, and I hope we can see that. That's the U2S cell. Resolution is well below 10 nanometers in 3D with these recordings here. The colors are the colors encode the Z position, and what we can see here in this U2S cell is that the Mycos localizations they seem to be in clusters, but clearly they are not connected, and it's very difficult to tell whether there might be a spiral-like structure in here. I'm not sure because our brains are wired to see structures. So I personally wouldn't be convinced that we do see here anything. A computer wouldn't find anything, I guess. When we look to the COS7 cell, then it looks a little bit different. Here, the Mycos clusters clearly seem to be, at least in parts, more closer connected. And for me, at least here on my monitor, it's very clear that it seems to have a kind of a spiral-like structure. So if you analyze this data in a different way, and that is that we try to project all the Mycos clusters into a plane. So what we have been doing here is we have taken, say, for example, this mitochondrium here. We took it to the computer, then we opened it up, and so that in this projection here, the top of the mitochondria and is in the center, and the bottom here is at the left and the right, and we see here in every individual cluster. We also took into account that it's a bit spiral-like, the entire mitochondrium, and that there are length variations. But if you correct for that, we get to these patterns, or to these distributions of the Mycos of the MIC60 clusters. And of course, that is something we can analyze. I do not want to go into the details, just keep it here with the visualization, but it's clear. Here we do have individual clusters. We don't see any pattern, whereas here on the right-hand side in the COS7 cells, we do indeed see these arranged Mycos clusters, which are in stripes. There are clearly some patterns indicating that in this very cell line, Mycos is arranged, and this is some of the analysis we at the moment are trying to explore. That is, everything is here very at the beginning, and let's see how far one can push that. And I'm very curious how the community, we as a community as a whole, will explore these highest resolution technologies in the future to understand mitochondrial architecture. So this is a kind of a loose end, and therefore I would like to, in my last part, my last 10 minutes or so, I would like to change gears a bit, but keep with the Mycos complex. So far we have looked to the Mycos complex as a player in maintaining overall mitochondrial network morphology, as a player required for tubular mitochondrial formation. But in fact, Mycos has been identified mainly as a player required for maintaining or generating the architecture of the inner membrane. If you delete the entire Mycos complex, then we no longer have this nice arrangement of cristae, but we have a different aberrant architectures. So we decided to look into the role of this Mycos complex in more detail, because if one understands its role better, one might be able to understand mitochondrial cristae biogenesis better. So what we did is we deleted all known 7 genes encoding proteins of the Mycos complexes in HeLa cells, and via CRISPR-Cas, and analyzed all of them, all cell lines. I don't want to go into too much detail here because it's really for mitochondrial aficionados. But what you can see from the overall structure is that the MYC60 has a very strong phenotype, as does the MYC19. They form, as I told you previously, a subcomplex. The MYC10 knockout also has an effect on the overall network morphology, although it's less strong. When we look to the Western blot, what we find is that if we delete the MYC10, then indeed, of course, the MYC10 is gone, also MYC16 and also MYC26. So the main components of the MYC10 subcomplex are gone. However, MYC60 and also MYC19 are unaffected. That means if we delete the MYC10 subcomplex, we still have an intact MYC60 subcomplex in these mitochondria. If we delete the MYC60 in these cell lines, then the entire Mycos complex is gone. So that allows us to disentangle the function of the MYC60 subcomplex in the delta MYC10 cells, whereas the entire Mycos complex. So we decided to look a bit more into the morphology. As I told you, was very well known by many groups that the cristae architecture is strongly affected, and of course you also saw that. Here, that's the wild-type architecture that are electron tomograms reconstituted. They have a depth of around 200 nanometers or so. Here are the lamellar. We do find differences between the MYC10 knockout and the MYC60 knockout. These are just typical examples. In the MYC60 knockout, the cristae often look like onions. So we have tubes of inner membranes which are within tubes. So here would be the most inner tube, and then there's a second tube, maybe a third tube, and then here in gray, that is the inner boundary membrane. There are no or almost no cristae junctions in these MYC60 knockouts. The situation is a bit different in the MYC10 knockouts. Here, typically, we have only one tube of inner membrane. In gray, again, the outer membrane, and then here in blue, that's the inner membrane. However, we do find cristae. That was a bit of a surprise because it has been previously reported that there are no or very few cristae junctions, not cristae junctions, in these mitochondria. They are there. The number is reduced, and also the shape of these cristae junctions is a bit different. They are more roundish, whereas in the wild type they are more lengthy, and also the localization seems to be a bit different in these knockouts, but we clearly have them here. So we can conclude from that in these knockouts, that we can conclude from that data that even in the absence of MYC10, we do have cristae junctions, but then when the entire MYC60 complex is gone, we don't have some. So we conclude that we do need the MYC60 subcomplex for cristae junction maintenance. Well, what we then did is we re-expressed in these knockouts MYC10 or MYC60, and we did that by generating cell lines which under an inducible promoter allowed to express the MYC10 or the MYC60, and then we looked to the repair of these mitochondria. And the reasoning was when we can understand how or we come back from this very aberrant mitochondrial structure to the healthy structure, we should be able to learn a bit on how cristae might develop and how dynamic cristae might be. Because of the limited time, I just want to give a few data on the work on re-expressing MYC10 in the MYC10 knockout. And if you do so, here's the data. First of all, the light microscopy data. That is this MYC10 knockout cell line, which can be induced to re-express MYC10 flag. Then we see that after 16 hours, most cells start to express the MIC10 flag, and after 24 to 2 days, then we do have almost close to normal levels of the MIC10. Again, if you look to the Western blots, one can see the same thing as we see here on the left-hand side in the images. At the start of the induction, we have basically no MIC10 flag. Here, this band here is presumably background band. And then after 8 and 10 hours, we start to get large amounts of the MIC10 flag. And interestingly, when we express the MIC10 flag, also the other components, such as MIC13, are then stabilized, and we can co-RP them together with MIC60 in an entire hollow Mycos complex. Of course, MIC19 and MIC60 are unaffected because they are all the time there. So we can look to the microscopic data. And we see here and ask the question, do we have, when we re-express the MIC10, do we have a repair of the mitochondria, or do we observe the mitophagy, that is the degradation of the aberrant mitochondria, and then the replacement of the aberrant mitochondria by new mitochondria? And all our data seem to indicate that we do have a repair. And I just show you one data set here. That's an exemplary data set, of course. We have here the knockout situation, the outer membrane here, the inner membrane. This is this tube-like arrangement. And then after 16 hours, we typically see this intermediate mitochondria. Here are some lamellar, already some lamellar mitochondria, whereas here in the middle, we still have this aberrant typical MIC10 cristae phenotype. This is just a blow up where you see here this tube-like inner membrane, which is typical for with the MIC10 knockout. After 24 hours, the mitochondria look mostly wild type like, and of course one can do quite a number of analysis here. And one important piece we saw is that we have very good data that we have the development of secondary cristae junctions, as we like to call them. So that are cristae junctions which develop on aberrant inner membrane sheets. I don't have the data here. That's most notable on the MIC60 knockout, where when we re-express the MIC60 knockout, then once in a sudden cristae junctions develop on aberrant cristae. So that is again a very good indication of repair and not of replacement of the mitochondria. So of course one can also look to distributions of mitochondrial proteins upon re-expression. And this is shown here. Here we are looking to the distribution on the left-hand side of MIC60 in these cells which are deficient of MIC10. And if you look to this blow up here, what we see are again these two-sided opposite distribution of MIC60 in this MIC10 knockout. So what that means is if you delete the MIC10, then once in a sudden, we indeed have again these opposite distributions. My computer is getting low on power, just a second. So here we are. Yes, I don't know. I hope that works with my computer. Give me a second. Give me a second. This is strange. It's plugged in, so let's hope for the best. Right. We do see here a two-sided distribution. So by deleting MIC10, we can induce these spiral-like, or this pattern distribution of MIC60 I was speaking about before. When we now re-express MIC10 in these cells, then we get a redistribution of MIC60. It starts as a more spiral-like pattern, and when the mitochondria are looking normal again after 24, 48 hours, then there is no structure again, but the cristae are looking normal. And of course, one can do that kind of analysis with super resolution, looking to different proteins. And this is what we currently are doing with a lot of different proteins, trying to understand how proteins move in mitochondria when the cristae are getting repaired. My time is over. So I just want to show you here some FIBSEM data on the repair mechanism, because that allows us to give us an indication of how we think mitochondrial biogenesis might work. That here is a 3D reconstructions of FIBSEM data. We do see in blue only the cristae membrane. So the transparent part is the inner boundary membrane and the outer membrane. So we are looking just in the interior of the mitochondria. And you may be able here, so this is a kind of a tubule within the inner boundary membrane, and we may even be able to, when it moves here from the side, to look through this entire thing. I guess one can see it from that side better. Here one can see it from the bottom one. So that is the knockout. Here is the wild type situation, the very nicely stacked lamellar cristae. And here, that is one of these immediate mitochondria, and now in 3D, and we can clearly see this undulation of the cristae membrane, which starts to connect itself, for example, here or here, with this gray part, which is in a boundary membrane. So we believe that this inner membrane here is just starting to fold up and to be repaired into cristae. So what kind of model can we take from here? And I would say it's the following. Our idea, and there are many other interpretations, but that is our current idea, is the following. When we repair the Mycos knockout, we first start with an invagination, and then it grows. In the Mycos mutant now, it would grow for very long times, because it wouldn't be able to reconnect with the inner boundary membrane. In a wild type situation, we probably have the same start, but then this inner membrane here might connect via a secondary cristae junction, and we know they are existing, and there's a possibility to develop them. And then we might have an undulation of the inner membrane. We know these cristae membranes are highly dynamic. They can fuse at the other side of the mitochondrium with the inner boundary membrane, make cristae junctions. Then they can divide, and there's good data by other groups, for example, by Andreas Reichert's group, that there might be fission of cristae. And then once in a sudden, we have this group of cristae here. And of course, these things might be very dynamic. But that one might be a way how one can develop with very simple modifications of the inner membrane, these cristae. And of course, they could be easily converted into lamellar cristae or tubular cristae. And by the way, because this is a group of cristae, I would propose that the cristae groups you have seen in the lifestyle movies at the very beginning, which are interspaced with nucleoids, might come from just one event to generate these groups. So with that, just a second view on that, on how we could believe these groups of cristae might develop. I'm at the end, and I'm coming to the most important slide. I would like to thank people. In my team, most of the data I've shown today has been generated by two extremely talented postdocs, that is Stefan Stolt and Stefan. Quite a few of the people in the lab contributed. Mark Bates, he takes all the acknowledgements for the 4-pi storm data. Of course, you have many collaborations with Stefan Hell, and they pay the bills. And well, thank you for the attention, and I'm very happy to take questions.
Wonderful. Thanks, Stefan. That was awesome. There's one question straight up from Tan here. He's asking, how do you think MIC60 promotes the growth of the inner membrane, and what would be the source of the lipid for that growth?
Lipids will, I mean, I'm not entirely sure whether MIC60 is required for the growth, because when you delete MIC60 or the entire Mycos complex, we observe excessive inner membranes. You know, we have these layers of inner membrane which forms this onion-like structure. So my take here is that MIC60 is not required for the development of inner membrane. Lipids clearly are super important, and I'm very sure that and there's a lot of data on that lipids are required for determining also the inner structure of the structure of the inner membrane. The question probably is what limits the growth of the inner membrane, and unfortunately I have no answer to that. That's a very exciting question, because at some point obviously the growth of the inner membrane stops, and what the mechanisms are not known to the best of my knowledge. But I'm not sure whether Mycos is a major player here. I guess its major role is indeed in the maintenance of the crystal junctions.
And there was one more from Jennifer Zenker. Jenny, do you want to speak?
Yeah, I hope you can hear me. Stefan, that was super fascinating. Thank you. I'm really curious, is there anything known if these MIC proteins play any role during early embryogenesis, where you literally start with very small globular mitochondria that have no cristae at all, and within few days they form these cristae and then become tubular, like, well, a tubular network?
That's an awesome question.
Thanks.
And I do not know. That is definitely something worthwhile to look at. I cannot tell. I just do not know whether people have looked into that. I would guess it has been done, but to what detail, I just don't know.
Okay.
I know that there's some fly work on this.
Okay.
And that in flies, people do see different expression levels and also different distributions of the Mycos. I'm not sure how much has been done in human cells, but it's something which is, you know, my ignorance.
Okay. No, thanks. Maybe I will see if I can find anything, but if you come across anything, I would love to know. But yes, thanks.
Absolutely. I will have a look. That's
awesome.
That's an awesome question.
Thank you.
We've got one from Peter Curie here. How do the beautiful shapes and forms of mitochondria in the cells that you show relate to differentiated cell types in vivo? And do you have any ideas how this dynamics is regulated in living tissue?
Well, again, it's an awesome question. And we know that there is a relationship between the architecture and the function. For example, we do see in cardiomyocytes, we do see highly stacked lamellar cristae. And we do see that in other cell types, we have different architectures. Really, to the best of my knowledge, there is no really very clear evidence how these structures relate to function. And we have all these anecdotal evidence, but I'm not aware, at least I am not aware of a really systematic analysis of shapes and function beyond anecdotal data. What we know is that expression levels of, say, MIC10, or the relative expression levels of MIC10 and MIC60 vary. There's a variation. And also OPA1 seems to be an important player in different cell types. And that might have an influence on the architecture. But at the end of the day, the regulation of cristae architecture is still a really unexplored area. So again, I have no good answer on cristae architecture and tissues and how that is regulated. That's certainly something which is super interesting to explore.
There's another one from an anonymous one, which is probably following on from that too. They're asking about how the cytoskeleton and motor proteins might be contributing to this process of, I assume, the cristae repair. But then, I mean, also it could be contributing to the twisting.
Yeah. I mean, what we know is MIC60 is, of course, a promiscuous protein. It's considered kind of a platform to interact with very many other proteins. And there's one very, very interesting connection, and that there seems to be a physical connection to mirror and the motor proteins that are required or that are mediating the trafficking of mitochondria along the microtubules. So that gives kind of ways to the kind of interesting or tempting idea that a way to move mitochondria in a cell is that the motors indirectly are connected to MIC60 and thereby to this potential larger network in the mitochondria, which might form a scaffolding structure, and by that they could move the mitochondria through the viscous cytosol. That I think it's a very, very interesting idea that one might follow up. So there is clearly evidence that there's a physical connection of the Mycos complex to components of the microtubule cytoskeleton.
And I think we've got time for one last one from Mike Ryan. If you want to do that, Mike, or do you want me to read it out?
Oh, that's great. Thank you. Stefan, lovely talk. Beautiful work. Stefan, it's related to that last question. And we know that the MIB Mycos is there, and the SAM machinery is part of that interaction with MIC60. Do you see a similar type of spiral formation with the SAM complex that correlates with the MIC60?
Hi, Mike. The SAM, occasionally we had the impression. The problem here is we have no good antibodies against the SAM at the moment. And that really restricts our possibilities to visualize it. So we are waiting here for genome edited cells that would allow us to to flag tag it and then to look at that. I would expect to have also such a spiral-like arrangement with the SAM complex, yeah. But we have no good data on it.
And Georg, you've got one in just in time. Georg Raum is asking, can you correlate the presence of MIC60 with the diameter of the mitotubules?
Can we?
Where are you, Georg?
Yes. I mean, there's a correlation, and the correlation is if we have if we have very thin mitochondrial tubules, we tend to see this opposite distribution of the MIC60 better than in larger ones. There seems to be a correlation. I mean, as I said in the beginning, this opposite distribution of the MIC60 is not always there. It's very much depends on the cell type, and we believe that we see it better when these bands are very narrow. When they widen, then they move around the mitochondria. We don't see it anymore, and it seems that in larger mitochondria, these proposed bands are wider, so we don't see them anymore, or not as good. So there seems to be a correlation of the distribution of Mycos and the diameter of the mitochondria. Although, I mean, it might be different for different types of mitochondria, but there's a general idea along this line. Yeah.
Yeah, just sort of in the initial one of the initial images we show in I think the live cell super resolution, you see the cristae, and then there are the sections where there's no cristae present, where the mitochondrial DNA is. And obviously where the cristae is the MIC60. Then in the sections that don't have the cristae, are these tubules wider than in the areas where you have the cristae? And I don't think that's
no.
Yeah.
And I'm not entirely sure whether you're right that the Mycos complex is indeed only at the cristae junctions. I mean, there's good data that MIC60 is enriched at the cristae junctions.
Okay.
But it's not entirely sure that it's nowhere else, and it's certainly not, proven that the MIC10 is just only at the cristae junctions, I guess.
Okay, thanks.
All right, well, I think that's our time up. So it just remains for me to thank Stefan for a beautiful talk, for giving up his bright and early morning. That was fantastic. Thank you so much, Stefan.
It was a pleasure. Thank you. And if anybody's interested in working on this area, give me a call. We have positions.
Fantastic. Awesome. All right. Thanks, everybody.
Goodbye.